Okay, awesome. Thanks. Okay, so yeah, so the point of the research and action series is to bring together MSU researchers, K-12 educators with that very specific focus of thinking about the classroom experience in science. So really thinking about how the stories of MSU STEM researchers can connect how we approach our science learning experiences and think more. Yes, we want to talk about the content and how our content relates to this in the moment research that's happening, but also how the stories of researchers can change the way we consider student experience in the classroom. So in the chat box, if you don't mind, we're going to take a moment to get to know each other. And I'd like to hear your name, um, a subject you currently teach, or your favorite subject to teach, with your location, ideally, and then a time when you knew you could do science. So you knew that you were able to do science. So I'll leave that open, and if you could pop it into the chat, that'd be great. I'll put it in myself too. Okay, so I just put in mine, Jeannie Chips. I actually taught high school chemistry mostly, but I taught a little bit of high school biology and some middle school science classes. I had a unique experience as a kid because my dad was a high school chemistry teacher, so I feel like he often brought home science experiments. I definitely remember playing around with dry ice as a kid. He brought liquid nitrogen home. There were all sorts of experiments that we did in our kitchen and in the backyard and sometimes in the front yard because we launched bottle rockets a lot. But I remember designing a perfume making experiment when I was in third grade, I want to say, and that was the first time I was like, I can do this science thing. So, ooh, Laurel worked in marketing for a surgery center. So cool. Love it. Oh, I love that. Yeah, I'm not gonna lie. So right now I work with undergraduate pre-service teacher educators, and I talk a lot about how I'm so grateful that I was a science teacher because there were so many hands-on inquiry activities where you could do experiential learning so easy. And yet, let's just do this. Y'all are gonna design things, and I'm gonna make sure this is how I support you. So a water testing field trip in eighth grade. Esther, I love that because I used to do so many water testing field trips with my high school students. So I love to hear that that like made you feel like you could do science. That's awesome. Awesome. And then Tom, yes, growing up on a farm. Yes, thank you. There's so much science happening on farms that, yes, bringing in that idea that like we're just doing science by experiencing the world. I love that. Nice. And I think it's awesome that so many of you ended up in the science fields. I have one brother who is a PhD. So my dad was a high school chemistry teacher. The middle brother, he has his PhD in chemistry and is running a lab now. And the oldest brother, he rebelled. And for the longest time, he stayed away from science. But now he runs a company that does like um, environmental quality monitoring systems for like asbestos, lead, a bunch of other chemicals. So we all kind of ended up going there too. So Tom, I love that. Thanks for sharing, everyone. Okay, so we're going to just do a quick intro to what is STEM self-efficacy, because it's really going to hopefully situate the rest of our conversation together. Um, and for some of us, you might have started to feel like what self-efficacy meant as we were talking through that just now. But really, STEM self-efficacy, it reflects task-specific performance expectations. So things like, I believe that I can get a B or better on my math test. It is not inherently evaluative. So it's not like I am good at or I am bad at instead of I can do, right? So when we were talking about I can do science, it's really whether we feel that we have the capability. It's not necessarily comparing to others, like I can't do science as well as my friend can do science, or I do science better than my friend does science. Instead, it's really on like, I have mastered this ability to identify a variable, to do this titration task, right? And it's very, um, it can be very minute where you feel very you have a sense of self-efficacy about a specific science. And this happens a lot for different learners. And this is where it's important to understand that STEM is a broad term where we do have science, technology, engineering, mathematics. You might have a strong science self-efficacy and a slightly weaker mathematics self-efficacy, and that might influence how you perceive yourself as a STEM person. So self-efficacy is really interesting to think about how it leads to our identity and where we see ourselves going in our future careers. So we'll come back to this in a little bit, but I'm going to hand the floor over to Esther. Esther is a doctoral candidate in chemical engineering, originally from Idaho Falls. She graduated here at MSU with a BS in chemical engineering and biological engineering. And so now, under the mentorship of Dr. Stephanie McCullough, Esther is going to be researching how to make more definitive and accurate diagnostic tests. And I'm not going to read the rest of it because I'm just going to cede the floor entirely over to Esther and let you pick up. I will stop sharing my screen actually so that you can share yours.
Great. Well, I think that should be, yeah, there. Okay.
Yep.
Yeah, thanks, Jeannie, for the introduction and also for inviting me to talk today. This is fun. Um, so yeah, self-efficacy in STEM. Um, and oh, I guess I'll say, yeah, I'm a PhD candidate in chemical engineering. I'm in my 5th year now, and hopefully maybe a year and a half until I graduate and finish the program. So when Jeannie gave me the list of topics that I could pick from, self-efficacy stood out to me because I hadn't really thought about it before and didn't really even know what it meant. So it's like, that would be a good one to learn more about myself and think about how that kind of played a part in my own development as a scientist. So I pulled some quotes from a couple papers about, yeah, STEM or self-efficacy is about having strong positive beliefs that, oh, I gotta move my box here, that you have the capacity and skills to achieve your goals. So like Jeannie said, it's really kind of goal directed and task specific, that you know that you have the capability to do a specific thing. And then self-efficacy influences the choices individuals make in terms of goal choice, the effort expended to reach those goals, and persistence when difficulty arises. So from these 2 areas, I kind of pulled out 3 things. This strong positive belief, kind of I interpreted that as building confidence in your capabilities, and then setting goals, like what goals do you choose and how do you reach those goals, and then persevering, this persistence when difficulties arise. So I've broken up, yeah, am I talking to those 3 areas and kind of how from high school into now graduate school, how those 3 have sort of played a part here. So I'll start with building confidence. So the same Rittmeyer and Breyer paper talked about building confidence in three different areas, that there's kind of these three different things that can happen to help you build your self-efficacy. So social persuasion is sort of the feedback and support that you get from the people around you. And then vicarious experience is learning from observing other people do something. And whereas mastery experience is actually the hands-on, like you actually do the task and gain confidence from doing that task. And I thought this quote was kind of interesting, how they break apart here that the most influential source of STEM self-efficacy for boys and men is this mastery experience, whereas the most influential source for girls and women are the vicarious and the social persuasion. So as a woman in STEM, I thought that was kind of interesting to think about how those maybe played a bigger role in my decisions to become a scientist. So in high school, so yeah, I'm from Idaho Falls, and there's a lot of engineers in Idaho Falls because they all work out at the Idaho National Lab. But my parents are actually teachers, they're elementary school teachers. So initially, engineering was not even on my radar of a career choice. I was actually kind of considering being a teacher. But then I had a lot of social persuasion, I think, in high school, where I had really good teachers, but it was actually my lacrosse coach who I think initially told me that I should be an engineer. And she worked out at the National Lab. So she was kind of telling all of us that, that they needed more women out there. And then I had a friend's mom who signed me up for this engineering state at Utah State University. And she was also a really important person in terms of giving me feedback on my capabilities as a scientist. And that she saw that in me and said, hey, you should do this camp, was really kind of a pivotal moment in my decision to be an engineer, especially because I didn't even know that engineering encompassed things like biomedical engineering and biological engineering. And it wasn't just all like motors and roads and bridges. So this engineering state went to Utah State and stayed in the dorms and toured labs and got to see college students actually doing their research. And one thing that I remember really clearly was seeing these biological engineers who were isolating spider silk proteins from genetically engineered goats. And that was, I think, a vicarious experience for me to observe this kind of crazy experiment and to just think, wow, that people do this stuff. Like other people are doing this, maybe I can do this same thing. Was really important. So I decided that I would study engineering and go into like biological, biomedical engineering. And I applied for college and went to MSU. And here also, I think, I mean, school and studies, there's a lot of mastery experience part in here too, but I also think that social persuasion and vicarious experience played a really large part in my undergrad throughout those 4 years of building my self-efficacy as a scientist. And one specific example is my friend Shauna here, who's in the middle of this picture. And she was the one who told me that I should do undergrad research with her in Dr. Connie Chang's lab. And Dr. Chang is on the left here. And I think without my friends stepping in and telling me, you should do this, I don't think I would have been brave enough to approach Dr. Chang myself. But then once I was in, Dr. Chang was a really important mentor there too, in giving me good feedback on my experiments and helping me kind of understand what research was and how to do that. So yeah, I graduated in 2017. And by that time, with these experiences in Dr. Chang's lab, I had decided that I wanted to go on and be to go to grad school and study more specific biomedical things. But before I did that, I wanted to take a gap year and try a couple of new things. So I worked for this company down in Peru called Alimenta Alga that did, they were in food. So they were trying to grow spirulina algae as a nutrient supplement. And I spent 4 months down there growing algae in 5-gallon buckets, basically, and doing micronutrient experiments with them. And this was a really important mastery experience for me because I hadn't had any internships before this point. I had just done, I had the research in Dr. Chang's lab, and then I'd done some study abroad stuff, but I didn't have any actual job experience. So kind of getting out of the classroom and applying everything that I had learned in a setting where my boss just said, hey, solve this problem, and then I had to figure out how to solve it, was a really, really important point in my self-efficacy development. So yeah, I think throughout high school and my undergrad career, there was a lot of just building confidence in myself as a scientist and as a person in STEM through these 3 different areas. So I thought that was neat how this paper had divided them up. So after Peru, we'll move into number 2, setting goals. And the literature talks about this sort of reciprocal relationship between setting a goal, completing the goal, and then having a boost in your self-efficacy after that goal is completed. And how then that can lead you to set even higher goals the next time. And this works as long as your capabilities and your goals kind of align and you have sort of a realistic sense of your self-efficacy. But then I think that you can get out of this cycle if maybe you set a goal, but you fail to complete that goal, and then your self-efficacy takes a hit. And so here, maybe you end up in kind of this limbo situation where your trajectory to set another goal is sort of in question, like, oh, can I actually do this? Are my capabilities up to completing this task? And so at this point in my development as a scientist, after my internship in Peru, I think I came across this this question mark period. So after I was in Peru, I came back to Idaho and I worked at this camp in the Sawtooth Mountains. That I'd grown up going to this camp, and I'd always kind of wanted to work a winter there and drive this snowcat around and shovel a lot of snow. And I was really excited to do this, but I also had this goal, because remember, I graduated and I was like, I'm gonna go to grad school, I'll just take this gap year off. So I also had this goal in the back of my mind that I needed to apply to grad schools and keep that part of my life moving. But I didn't set up a very good plan for how I would do that when I came back from Peru. And so these deadlines were passing and I was just in the mountains, not really thinking about it. And I ended up only applying to one grad school and not getting any funding to go there. So I had set myself this goal of going to grad school, but I didn't have a plan and I failed to like fully flesh out those tasks that I needed to do. And so I reached this point where I had to really come to grips with the fact that I had failed at this goal and that I didn't have the funding to go to grad school. And I ended up in this question mark area where I was thinking, should I even go to grad school? Should I just get a job? Am I cut out for grad school if I can't even manage to apply successfully? But fortunately, at the same time, I think life just intervened, and I had started dating my now husband, and he was living in Bozeman still. So I thought, you know what, maybe I should go back to Bozeman, go back to Montana State, and talk to those people who had been so supportive of me during my undergrad and kind of get their advice on what I should do. So I talked to the department head at the time, Dr. Jeff Hayes, and he was very much like, oh, don't worry, you can still apply and come back here if you'd like to, even though I missed the deadline. Um, and then I also talked to Dr. Stephanie McCullough, and I think she jumped um, on the call here. So yeah, so I talked to her, and I'd had I think two classes with her as an undergrad. So I had some background with her and knew that she was in more biomedical research, and that's what I wanted to do. And so just, and I knew that kind of the type of teacher that she was and the mentor that she was, and that I felt very comfortable with her and that I could really learn a lot from her. And she was awesome and kind and very much like, yeah, join my lab. So she's Stephanie, in her lab, she does microfluidic research and disease diagnostics with looking at different RNA and DNA protein things. So that's kind of how I ended up back in Bozeman for grad school, which wasn't the worst thing at all. I think it was actually a really great thing, but just kind of interesting for me to reflect on how this goal setting aspect of self-efficacy is not always linear, because you're not always going to complete all the goals that you try and set for yourself. So I think it is really important to remember to surround yourself with people who can give you kind of an outside perspective and maybe show you a path forward that you didn't initially see. And that can kind of put you back into that cycle of setting good goals, reaching those goals, boosting your self-efficacy. So now that third aspect of persevering, and here's where I'll get into my research and what I'm doing in Stephanie's lab. And I liked thinking about this part of persevering because I think this is kind of where developing self-efficacy as a student and as a scientist, kind of where the rubber hits the road and when it's super important. I got another quote for you. As one female engineering major put it, confidence that you can do this, that's what you need to persevere in the discipline. So knowing that you have people who can give you good feedback, and that you have these mastery experiences, that you've accomplished hard things and you've set hard goals before, and you've built this self-efficacy, then when things get hard or don't go the way that you think they will go, you can still persevere through that. So this picture here is one of the instruments that I've worked with the longest in the lab. It's an surface plasmon resonance machine that's used to measure binding kinetics of different binding pairs. And this has been very much a test of perseverance over the last 3.5 years. So I'll get into what I'm doing with it and why. But first, some background on my research in the lab. So yeah, my research area is broadly disease diagnostics and kind of trying to figure out how we can make better diagnostic tests. And diagnostics are an important part of the treatment of any disease, because you need to know what you're fighting before you can effectively treat it for the most part. And in the United States, there's a 10 to 15% misdiagnosis rate, and that translates to between 40 and 80,000 patients every year who are injured or die in the US due to those errors. And then on top of that, patient cost, there's also a time and a resource cost to wrong diagnoses, where you are wasting time trying to figure out what's going on based on maybe some really ambiguous symptoms. So it's important to get a good diagnosis fast. And this comes into play in lots of different areas of disease, like cancers, infectious disease, like we've seen most recently with COVID, and then also neurodegenerative diseases. And this is where my research also is focused, is in Alzheimer's disease. We have a collaboration with Dr. Anya Kunze in electrical engineering here at MSU to study different biomarkers for Alzheimer's and how we can detect those better. So the ideal diagnostic test kind of would have three main aspects to it, where you want something that's accurate and fast. You don't want to have to wait months to know that you have COVID, which fortunately we don't have to with current tests, but as an example, you want something that will tell you what's wrong quickly and accurately. You also want a test that is sensitive and specific. So this means basically that you can detect a low biomarker level. So the biomarker is some molecule that tells you that you have the disease. Maybe for COVID, that's messenger RNA. So you might want to detect really low levels of that so that you can catch the disease early. And then you also want to detect the right biomarker. So that's the specificity of your test. And then you want something that's reliable and economic. So you want it to work consistently every time, regardless of who is performing the test. And you don't want something that's super expensive. Another thing we talk about with diagnostics is whether or not it's point of care. So whether we can use it at the patient's location or close to where the patient is. And this is important, or often our mind goes to like a developing country where there's no electricity, or you're out really far away. But this is also applicable, I think, in states like Montana, where there's a lot of rural communities, and traveling to a large hospital or a lab is maybe not feasible for everybody. So it would be really nice to be able to just take that test to them. So an example is a pregnancy test, because it's pretty fast, like, yeah, a couple minutes, and very accurate. There's different levels of sensitivity of some of the tests on the market, but they're pretty sensitive, and they're really cheap. So that's one example. So in our lab, we're specifically working with nucleic acid biosensors. So we're trying to detect some disease biomarker using a different combination of nucleic acids. So nucleic acids are DNA and RNA, and these biosensors have four main components to them. So the first is the target, which can directly be a disease biomarker, or it can be something derived from your disease biomarker. So it can be different variety of molecules. Then the second one is the receptor. And the receptor is what is designed by the scientist to react with your disease target. And this, for nucleic acid biosensors, it's these different nucleic acid machines, basically, that can react in different ways. My research is mostly just with these complementary DNA. So leveraging over here these Watson-Crick base pairings, where the A's and the T's bind together and the G's and the C's, and designing something that will complementary bind with the disease target. And then the third part is some sort of signal amplification, especially if the amount of target that you're looking for is really small. You might need to add in a step that will amplify your final signal so you can actually see it. And maybe this looks like using a different nanomaterial, like a gold nanoparticle, or using different enzymes and amplifying, like creating more of your initial disease target, amplifying that, just the amount that's in there, creating more of it so that your signal is bigger. And then finally, you have the signal output. So one example of the signal output is a PCR curve that I'll talk a little bit more about here. But you have fluorescent dyes in your reaction that then, as you create more DNA, you get these signals. So you can see whether your disease target is present. Another example is maybe something that's a little simpler, like a colorimetric test. And this is more of a yes-no diagnostic, where in the top here, the negative COVID-19 ones are like red. And then if it's positive, it turns more purple. And I think this test, I think they're using gold nanoparticles in a certain way to kind of create that color change. Yeah. So yeah, the gold standard nucleic acid biosensor is polymerase chain reaction. And this has been around for quite a while and is pretty predictable. And basically there, you take the template DNA, which maybe in this case, we'll say that's the biomarker you're looking for. And there's a denaturing step at a high temperature to pull it apart. And then you have these primers that are specifically designed to bind to your template DNA. And then you use a polymerase enzyme that will fill in the gaps there. So you end up over here with another copy of what you originally started with. And then you cycle through it again. So PCR is kind of, it's very reliably predictable in that each cycle you get another copy of what you started with. And the output, again, looks like this, the plot I've already showed you, where you can kind of pull out specific concentrations too if you have a standard curve in there. So PCR is really great because it's efficient and it's been established and predictable outcome, and it's used a lot, and it's very powerful in terms of detecting a lot of different nucleic acid targets. The downsides of it, though, it can be a time-consuming process. And then because you have to denature the DNA at a high temperature and then go back down to lower temperatures and keep going back and forth, you need a thermocycler to do that temperature changing. So you have to have this piece of equipment that is expensive and needs to be plugged in and have power. So it's not really applicable to point of care all the time. So in our lab, that's one of the specific things that we're trying to work around, is needing a thermocycler. So we're specifically studying isothermal amplification reactions. So isothermal means one temperature, but then we come upon the question of how do we open double-stranded DNA if we aren't thermally denaturing it by ramping up to that high temperature? And the answer is that we're using enzymes. So this is a schematic of one of the first part of the reaction that we're really studying in our lab. And Stephanie and some other grad students before I joined the lab developed this reaction. So in the first step here, there's the target DNA, and then this receptor DNA is what we've designed, where it's a looped piece of DNA that has two binding sites for the target. And the loop occurs because there's this complementary region in the middle here. So the target molecule can bind then to this toe-hold overhang that sticks out here, and then it will pop that loop open and expose that second binding site. So then we go into step 2 where we have the target bound, and we use a polymerase to do the same thing like in PCR, just fill that in. And then there's a nickase enzyme in step 3. And remember, we've designed this DNA receptor so we can kind of pick what's in there. And within the blue region, we can put in a sequence that then will tell the nickase where to cut in the DNA. So nickase enzymes cut at specific places. So we can tell our nickase to cut right here and release another target molecule. So each time that this goes around, we create more and more of these targets that can just sort of feed back into that loop and create more and more, just creating more DNA as we go through it. And what I didn't show here is that there's a secondary product phase where these targets can bind to each other and make more products. So basically the output of this reaction has 2 phases where there's an initial rise and then the secondary steep rise from that second product. and it gives a really high signal, and we can even see that um, just by eye. And a former grad student took this picture with her cell phone. So it's kind of a neat reaction. And then my part of the research comes in when we say, how can we make this better? How can we understand this reaction a little more and manipulate it as well for a specific outcome? So that's where I'm working on something that I'll call tunable kinetics. And the two questions kind of that we have in there are, how does the structural design of that looped DNA receptor change the speed of the reaction? So the kinetics kind of describes how quickly that reaction happens. So how does that receptor itself change those reaction rates? And then how does the reaction output depend on those kinetics? And can we manipulate those kinetics through the loop structure to give us an output that we want? And so pulling apart this reaction, when we start with our looped DNA receptor, if we put that into two steps, where we have the first target binding to the first site, and then the second target binding to the second site, this reaction is dictated by these kinetic rate constants, these Kas and Kds. And this is a dynamic system, so the DNA, each of these steps can go both forwards and backwards. So there's association steps, and there's a dissociation, or there's association rates and dissociation rates. So this is what we want to know. And this is where that surface plasmon resonance instrument picture that I showed you comes into play, because that's how we measure these Kas and Kds. So this is the most math that I'll show you. Basically, we can we can write these reaction rate equations using that are dependent on those reaction rates, and kind of calculate the rate of product formation based on these parameters. And why this is important is that literature has shown that having an increasing number of dependent binding sites causes a delayed start and a steeper binding curve in your final output signal. So in our reaction here, we have two dependent binding sites. So there's two red areas, and they're dependent on each other because of that loop. So when the first target binds, it pops open the loop and makes a more accessible second binding site. And this graph here kind of demonstrates how that signal changes. So as you're going from one binding site, and then the red line is kind of our situation here where we have two, but we keep getting this delay, but then this steeper binding curve. So if we really want kind of a quick change in the signal, maybe we keep adding more binding sites. And that's kind of where my research is exploring. And that's where the surface plasmon resonance instrument comes in. So I think I've got a picture of it up here again in the corner to remind you what it looks like. And how this instrument works is that there's kind of three components. There's a light source, and then there's a spectrometer that detects the light. And then in between those, there's a sensor. And the sensor has a layer of gold nanoparticles on it. And those gold nanoparticles, when the light source hits them, they emit what's called a surface plasmon. And that surface plasmon, there's a lot of physics that I don't understand either, but it creates this resonance peak. And then as you bind things onto the surface of the sensor, you can create a shift in that peak. And so in our experiments, we bind the receptor DNA. So I've got down here so the little Pac-Man guys are our loop DNA. We bind those to the sensor, and then we flow, on top of those, we flow the target molecule. So these little triangles are our target molecule. And as those targets bind to the receptor, we get a shift in the wavelength. And so the data collection looks kind of like this, where we get all of these different curves at different concentrations of those targets. And then we can put them all together into one graph And then that's where we use those equations that I showed you on the previous, or two slides ago, to fit a model for the data, in order to calculate those KAs and KDs and know those kinetic rate parameters. So to tie it back into perseverance, this is where I think research research is hard, and there's a lot of times where it doesn't go as planned. And so our journey with this instrument started back in 2019, and basically for almost 2 years we were troubleshooting, and we had to send it back to the company. We had to send samples to the company to get their help on things. We had to pull the instrument apart and see what was going on inside. So it wasn't until now, I guess it's almost another 2 years, when we finally were able to get good data. So I've been testing 6 different DNA receptors now, and we're finally at the point where I have one experiment left, hopefully, I think, and we'll be able to fit all of these curves to the proper model and be able to write a full story of what's going on with these different designs. And so, to conclude the research part of this, I won't get too much into the Alzheimer's, which is also interesting, but basically nucleic acid biosensors are useful to detect a lot of different molecular targets. PCR is the gold standard, but there's some drawbacks in terms of the equipment that's required. So we're really interested in these isothermal amplification reactions and how they can give us some advantages. But what we don't know is how we can tune the kinetics of those reactions for specific outputs. So that's kind of, yeah, that's the realm of my research, and hopefully we'll have a paper out here pretty soon on that part of it. So these are the same quotes that I showed you at the very beginning. And kind of to wrap up everything here, I think self-efficacy definitely, the development of self-efficacy for me as a scientist has not been a linear path, and it probably isn't really for anybody. But having sort of all of these aspects of building confidence and learning how to set goals and learning what to do when your goals aren't met, and then learning how to persevere through some really difficult, through more difficult times, like when your SPR instrument will not give you the data that you want. I think all of those things work together to help you understand how your capabilities can really help you reach the goals that you want to reach. And it doesn't stand alone either. Like, we're really shaped by the people and relationships we have around us. So when our own belief in our abilities kind of crumbles, knowing who to go back to and having those support systems is really important. Yeah, so that's kind of how I became an engineer and where I'm at in my engineering. And I think I'm over time here, so I will end there. Thank you so much.
Thank you. That was perfectly timed. So actually, that's great. If you can stop sharing your screen.
Yes.
I really enjoyed that science because I think we were talking at the Science Olympiad. Those isothermal amplifications, I'm so interested in. It's so cool to hear. Yes, get rid of some thermocyclers from an environmental perspective. I think that's really interesting. So many directions. I love the work you're doing. So thank you. I'll go ahead and share my screen again, and we are going to be talking about how to bring this to our students. So I'm going to go ahead and in the chat, I'm going to place a link to a Jamboard that will hopefully let you all start editing. And in that Jamboard, on the first slide, I'd love for you to like put a little post-it on what sort of curricular connections you can make. And it's so funny that the way you talk about SPR. I have tried to make SPR connections to chemistry for years because in IB chemistry, we have to talk a lot about um, some of the transition metals and the cool things that transition metals do in terms of color change. And what gold does, those gold nanoparticles are so unique and they're so cool and different. And if you all have not seen gold nanoparticles and what they do, there's really cool stories about how to connect gold nanoparticles to like stained glass windows or stained glass mirrors or windows and things like that. Super interesting. Love, 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 love that. That's the minute you showed the picture with the blue and the red, I was like, oh, I bet that's some gold nanoparticles too. Yeah, just those colors are so distinctive. So and it's mind blowing to students when you have them, when you give them those gold nanoparticles and they're thinking, oh, gold is going to be what we traditionally think of at the macro scale of gold. And so when they see blue and red, they get, what? What? That's gold? That's not gold. So if you can make those curricular connections on page 1, that would be awesome. And then on page 2, Esther gave us such a beautiful, rich description of self-efficacy and how it applies with your life. So thank you. I'd love for you all to think about, how can we use Esther's story to help to support our students. So that would be on the second slide. So if you could put those post-its. I love how much you talk about that interaction between individuals as a means to increase self-efficacy, because I really do think that's where us STEM teachers can play such an important role. So thank you for highlighting that. Awesome. Do you all have any questions about either the science or Esther's story? I know I have so many questions about the science. I could just keep going on and on, but I'm coming at it from a chemistry perspective. Laurel and Tom with middle school science and AP Bio. I mean, this whole thing, I'm sure, Tom, you were just like, oh, that's AP Bio. Oh, that's AP Bio. And again, the idea, I don't know, Tom, if you had difficulty getting PCR equipment. Like, yeah, right? Exactly. Could you imagine? I mean, if you had these isothermal amplification opportunities that were inexpensive and you could quickly do them with your AP Bio students, so many curricular connections I could think of there. We were a relatively old school, so we had some PCR machines, but they were from like the 90s, and I was teaching it in, 2018, 2019. Like that's, I'd love to have some more up to date ones, but we need equipment that's not quite so expensive for us high school teachers. I'm going to just click on the Jamboard link. Hopefully, I'm going to be able to see what you're all working on. You haven't used a Jamboard, over on the left, there's a set of controls, and one of them is called a sticky note. So I'm going to go ahead and do surface plasmon resonance micro scale. Here's how I might use it in chemistry. Human body systems and diseases. Absolutely, especially when you're talking about how do we detect diseases. I love that. And there's so many broad angles you could attach this to, right? Like so many different diseases. And then if you get a chance to go to the second slide to think through how is it that we can use Esther's story to increase student self-efficacy? Yes. Thank you for pulling out that quote about how, at least in that one research study, different genders were responding to different aspects of experiences that generated stronger self-efficacy. Yeah. Yeah, I completely agree. Students are definitely starving for this. And I think it helps by thinking through those three aspects that not everybody gets the same benefit out of a mastery experience, and not everybody gets the same benefit out of vicarious experiences, right? So you being a cheerleader might be particularly helpful to one set of students, but you also need to provide another opportunity for mastery experience for other students. So that is really interesting to think through. I love it. Okay, so I'm going to go ahead and attempt to get back to my presentation screen. Over here. Like, so some of the other ways that research suggests you can support students' self-efficacy is to track that and to help them track that self-efficacy with just these simple statements. So I pulled out another research article that looked at how researchers measure self-efficacy with students. And there's these really simple questions that could actually be quick exit tickets. Like, how confident are you that you could tutor another student? How confident are you that after you've read this little passage that we were reading, you could explain the main ideas? What I appreciated about this self-efficacy set of like measurements is that they tie very specifically to our content areas. So if you're measuring self-efficacy, you can get task specific, and it's kind of doing two things. It's helping you understand where is the self-efficacy levels of my students right now, and who do I need to maybe support a little bit more in their sense of like, I can do this. And also, if you go task by task within your content area, it's also going to give you a sense of this is what I need to teach, and this is what I think I'm good to move on with. So what's helpful about self-efficacy to me is that it really is coinciding very clearly with like my content area instruction. Some of the other suggestions are actually to reduce competition and comparisons, right? So if self-efficacy is really about oneself, when you're creating those competitions, oftentimes it leads to too much emphasis on those comparisons, and then it doesn't provide that ability to say, oh, I can do this, because you're making your emphasis is on comparing yourself to others instead of thinking about what you yourself can do. On the other hand, if you increase collaboration, you're going to get that, you're going to surround yourself with that system of we can do this together. And I've seen you do that, and I know you can do it, right? So these are some of the things research says that you can do. I want to first of all thank Esther so much for coming in tonight and for pulling out those quotes. Your quotes are really perfect. I absolutely love them. I think those quotes are incredibly beneficial to thinking about self-efficacy. If you have a chance, I am actually conducting some research on this process and how best to deliver this type of PD. So if you would not mind filling out a quick research survey, I will put it into the chat right now. And I would greatly appreciate that. Also, if you don't already receive our SMRC newsletter, please feel free to click on, I will put this link in the chat as well. We are going to be hosting some more research and action series in the spring. We're also going to be hosting a local in-person workshop. So I know Great Falls, you're pretty far away, Tom, so I'm not sure if you would want to come down. Shepherd Middle School, Laurel, I can't remember where you're at. I think you're up north if I remember correctly, but yeah, if you are interested, there will be, you're close to Billings. Actually, Laurel, then you might be interested, check out our website or the newsletter, because we're going to be offering that in-person workshop in the spring if you'd like to hear more. And who knows, maybe we'll get our hands on some cool nanoparticles and do some nanoparticle work. I would love to do that. I always like playing with nanoparticles. So, anything else? If you have any questions, feel free to stick around. But again, Esther, thank you so much for inspiring us, for telling us your story. I can't wait to hear more, and congrats again. I will stop the recorder too.